Good morning, my dear student. I am Dr. Kiran Ramrao Borkar. Now today I am going to discuss about the structure and function of pancreas. So, you can see here the picture of pancreas. This pancreas is elongated, light tan or pinkish in color. Tapered organ located across the back of belly behind the stomach. Pancreas has main four part, that is head, neck, body and tail. This head is the widest part of the organ and lies in the curve of duodenum, the first division of the small intestine to the small tube, and that small tube is called as a pancreatic duct. Pancreas is about 6 inches long. The pancreas is unique in that it's both an endocrine and exocrine gland. In other words, the pancreas has the dual function of secreting hormones into blood and secreting enzyme through the duct. Now you can see here another picture. Look at this. Okay, so predominantly the exocrine part consists of, it means that you can say here, when we take the TS of pancreas, at that time you can see here this pancreas has you can say mixed gland. Why it is called as a mixed gland? Because some part, that is, look at this portion, predominantly exocrine gland consists of grape like structure. Look at this grape like structure, and this grape like cluster of secretory cells that form a sac, and that sac known as a acini, which connect duct that eventually empty into the first portion of the intestine, and that first portion of the intestine is called as a duodenum. This acini, that duct, is opens into the small intestine part, that is duodenum. Okay, and the acinar cell shows peripheral clear zone and central zone containing zymogen granules, which discharge into the lumen during the period of secretory activity. The main duct of the gland communicates with the bile duct and discharge secretion into this duodenum through the sphincter of Oddi. Embedded within the pancreatic acinar tissue, there is a relatively small amount of the cellular tissue, and that is quite different in appearance, which occur in the lethal groups, and that is interalveolar cell, that is islete. It is also called as a islete of Langerhans. Look at this portion, this group of tissue is called as a islete of Langerhans, and this islete of Langerhans consists of three different types of the cell, that is alpha cell, beta cell and delta cell. Three types of cell are in islete of Langerhans. And pancreas involved in, when we see the function of this pancreas, at that time mostly you know that endocrine part is that the islete of Langerhans, and exocrine part is that the acini. So this pancreas involved in blood sugar level. Look at this, involved in blood sugar control and metabolism within the body, and also secretion substances. Collectively, that substance is called as a pancreatic juice that help in digestion. These are divided into endocrine role and another one is the endocrine role relating to the secretion of insulin and other substance, that is and other substances within the pancreatic islete that help in control the blood sugar level and metabolism within the body. And exocrine part or exocrine role relating to the secretion of enzyme involved in the digesting substance in the digesting tract. So we take first function, that is blood glucose regulation. What happens here? The cell within the pancreas help in maintain the blood glucose level, that is called as homeostasis. And the cell that this are located within the pancreatic isolate of Langerhans. Look at this pancreatic isolate of Langerhans that are present throughout the pancreas. When the blood glucose level are low, then at that time, look at this alpha cell secrete glucagon. Glucagon is also the hormone. Okay, this alpha cell secrete glucagon, which is increases blood glucose level. When blood glucose level are high, at that time what happens? This beta cell, this high at that time, this beta cell secrete insulin. This insulin hormone, for what purpose? To decrease the glucose in blood. Then third type of the cell, that is delta cell, which is also present in isolate of Langerhans, secrete somatostatin, which decreases release of insulin and glucagon. It means that you can say here somatostatin control both glucagon as well as insulin also. This glucagon hormone secretes that alpha cell act to increase the glucose level by promoting the creation of glucose and the breakdown of glycogen to glucose in the liver. It also decreases the uptake of glucose in fat and muscle. Glucagon release is stimulated by low blood glucose or insulin level and during exercise. Then second type of hormone, that is insulin. This insulin act to decrease the blood glucose level by facilitating uptake by cell, particularly skeletal muscle, and promoting its use in the creation of protein, fat and carbohydrate. Insulin is initially created as a precursor form called proinsulin, and this is converted to proinsulin and cleaved by C-peptide to insulin, which is then stored in granules in beta cell. Glucose is taken into the beta cell and degraded. The end effect of this is to cause depolarizations of cell membrane, which stimulates the release of insulin. Now we see the second function, that is digestion. The pancreas plays vital role in the digestive system. It does this by secreting a fluid and that contain digestive enzyme into the duodenum. This duodenum, the first part of the small intestine, that is duodenum, and that receives food from the stomach. This receives the food from the stomach. These enzymes help to, which enzyme? That is pancreatic juice, which is secreted by the pancreatic duct, okay, through the pancreatic duct. Then this enzyme help to break down carbohydrate, protein and lipid. This role is called the exocrine role of the pancreas. The cells that do this are arranged in cluster called as acini. This cell is called as acini, and secretion into the middle of the acinus accumulate in intralobular duct, which drain to the main pancreatic duct, which drain directly into the duodenum. Look at this, directly into the duodenum, and about 1.5 to 3 liters of the fluid are secreted in this manner every day. Every day secrete 1.3 to 1.5 liters every day. Okay. Then the cell in each acinus is filled with granules. cell in each acinus. Look at this acinus filled with a granule uh, containing the digestive enzyme, and these are secreted in an inactive form termed as a zymogen of proenzyme. When released into the duodenum, uh, they are activated by the enzyme enterokinase present in the lining of duodenum. The proenzyme are cleaved, creating cascade of activating enzyme. The enzyme that break down protein begins with activations of trypsinogen to trypsin. The free trypsin then cleaves the rest of trypsinogen as well as chymotrypsinogen to its active form chymotrypsin. Enzymes secreted involved in the digestion of fats include lipase, phospholipase, A2, lysophospholipase, and cholesterol esterase. Enzymes that break down starch and other carbohydrate include the amylase. These enzymes are secreted in the fluid in rich bicarbonate. Look at this. All these enzymes secrete into the this duodenum. And bicarbonate help maintain the alkaline pH from the fluid, the pH in which most of the enzyme act most efficiently, and also help to neutralize the stomach acid that enter the duodenum. Uh, Secretion is influenced by the hormone including secretin, cholecystokinin, then what is that? Uh, Cholecystokinin and um, VIP as well as acetylcholine stimulation from the vagus nerves. This secretin is released from the S cell, which form part of the lining of duodenum in response to the stimulation by gastric acid. Along with VIP, it increases the secretion of enzyme and bicarbonate. Then cholecystokinin is released from its cell of the lining of duodenum and jejunum, mostly in response to the long chain fatty acid, and increase the uh, effect of secretin. At the cellular level, bicarbonate is secreted from the acinar cells through a sodium and bicarbonate transporter that act because of membrane depolarization caused by the cystis fibrosis. Uh, then secretin and VIP act to increase the opening of cystis fibrosis transmembrane conductance regulator, which leads to more membrane depolarization and more secretion of bicarbonate. A variety of mechanisms act to ensure that digestive action of pancreas does not act to digest pancreatic tissue itself. These include the secretion of inactive enzyme, that is a zymogen, the secretion of a protective enzyme, that is trypsin inhibitor, which inactivates trypsin, the change in pH that occur with bicarbonate secretion and that stimulate the digestion only when the pancreas is stimulated, and fact that the low calcium within cell cause inactivated of trypsin. This is all about the digestion, and this digestion can complete with the help of pancreatic juice, because in this pancreatic juice most of the digestive enzymes are present, and it is very much important for the digestion purpose. So that's why we can say here the pancreas is a vital organ which play important role, that is it secrete hormones as well as uh, digestive enzyme also. Thank you so much.